Today we are going to talk about sexual reproduction in flowering plants. We can see a beautiful flower, but how does the sexual reproduction takes place in the flowers? It is by means of pollination and fertilization. We are going to discuss sexual reproduction in the flowering plants in detail. The sexual reproductive organ in angiosperms is flower. It is the morphological and embryological site of the sexual reproduction. The male sex organs are known as the stamen or the microsporophyll, and the female sex organs are the pistils or the carpel. Here we see a beautiful flower, and we can call it as an ideal flower with all the parts. See, this is the filament, this is the anther which comprises of the stamen, these are the petals, this is the receptacle, this is the stipple, and the stigma, the style and the ovary together comprise of the carpel. The fertile portion of the stamen is the anther. A typical anther consists of 2 lobes or 4 microsporangia, and such anther is called biathecous. In members of Malvaceae, anther consists of 1 lobe or 2 microsporangia. Such anther is called as monothecous and are bisporangiate. The long and slender stalk of a stamen is called a filament, and the terminal bilobe structure is called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in the flowers of different species. Here we see the 3-dimensional cut section of an anther. The pollen in the ripe stamen. When the stamen splits to release the pollen, we can see them, and the anther splits open to shed the pollen grains. A typical angiosperm anther is diathecous. The anther is 4-sided structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develops further and becomes pollen sacs. They extend longitudinally all through the length of an anther and are packed with the pollen grains. The anther wall consists of an epidermis which is single layered, and the function, as we all know, is protective. The endothecium, the cells of this layer, have alpha cellulosic fibrous bands arising from inner tangential wall, which helps in the dehiscence of anther. The fibrous bands are absent in the hydrophytes. The middle layer cells are ephemeral and are 3-layered. The innermost layer of the anther wall surrounding the sporogenous tissue is the tapetum. The cells of tapetum are nutritive, multinucleate, and show polyploidy. They are again of 2 types. The secretory or glandular type. These cells secrete sporopollenin, pollen kit, and compatibility proteins. Amoeboid or plasmodial or invasive type. Herein the cells undergo breakdown and the entire protoplasm move in the center and nourish the microspores. Here we see the structure of Microsporangium. It is the tapetum, the vascular strand, the epidermis, the stomium, the microspore mother cells. And this is where the microsporangium has burst and the pollen grains are seen to be coming out. The microsporangium is surrounded by 4 wall layers: the epidermis, the endothecium, the middle layers and the tapetum. The outer 3 wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost cell layer is the tapetum, which nourishes the developing pollen grains. Microsporogenesis, the process of formation of microspores from a pollen mother cell through meiosis, is called Microsporogenesis. The microspores are arranged in a tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Tetrads. These tetrads are of following 5 types: the tetrahedral, the isobilateral, T-shaped, linear and decussate. Pollen grains. The pollen grains represent the male gametophyte. It has a prominent 2-layered wall. The hard outer layer is the exine, made up of sporopollenin. It can withstand high temperatures and strong acids and alkali. No enzyme can degrade the sporopollenin. Pollen grain exine has prominent apertures called germ pores where the sporopollenin is absent. Here we see maturation of a pollen grain. This is a young pollen grain which has a haploid nucleus, and on undergoing mitosis, we see that a generative nucleus is formed and the tube nucleus is there, and the exine and the intine are very well differentiated. The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grains are mature, it contains 2 cells, the vegetative and the generative cell. It is spindle-shaped with a dense cytoplasm and a nucleus. In over 60% of the angiosperms, pollen grains are shed at the 2-cell stage. Now here's a problem. Even after killing the generative cell with a laser beam, the pollen grain of a flowering plant germinates and produces normal pollen tube because laser beam stimulates pollen germination and pollen tube growth. The laser beam does not damage the region from which the pollen tube emerges. The contents of killed generative cells permits germination and pollen tube growth. The vegetative cell has not been damaged, and the correct answer is that the vegetative cell is not damaged. The microspores, when mature into a pollen grain, the process is microsporogenesis, and next is microgametogenesis. In microsporogenesis, meiosis 1 and meiosis 2 takes place and a tetrad is formed. The gynoecium represents the female reproductive part of a flower. The gynoecium may consist of a single pistil, The condition is known as monocarpellary or may have more than one pistil. The condition known as multicarpellary. Each pistil has three parts, namely the stigma, the style, and the ovary. Here we see the structure and function of a flower. The pistil, which has the stigma, style, and an ovary. The stamen consists of the filament and the anther. Petal, corolla is there forming the petals and known as the whorl of petals. Pedicel and peduncle. Calyx is the whole of sepals. Here, more differentiated is the stamen, the anther and the filament, and this part is the stigma, style, ovary, which together comprises to form the pistil. This is the stamen. As you can see, the pollen grains inside it. The pollen is released. This is the ovule with two pollen nuclei and the egg. This is the pollen tube and these are the sperm cells. Finally, the ovary contains the embryo sac and this part is the ovule. Inside the ovary is the ovarian cavity, which is the locule. The placenta is located inside the ovarian cavity. Arising from the placenta or the megasporangia, commonly called as the ovules. The Microsporangium or the ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called hilum. The hilum represents the junction between the ovule and the funicle. Each ovule has one or two protective envelopes called the integuments. The integuments encircle the ovule except at the tip where a small opening called the micropyle is organized. Opposite the micropyle end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucleus. Cells of the nucleus have abundant reserve food material. Located in the nucleus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore through reduction division. Here we can see the conditions of apocarpy and syncarpy. The process of formation of megaspores from the megaspore mother cell is called Microsporogenesis. Ovules generally differentiate a single megaspore mother cell, which is abbreviated as MMC, in the micropyle region of the nucleus. It is a large cell containing dense cytoplasm and a very prominent nucleus. The megaspore mother cell undergoes meiotic division and the meiosis results in the production of 4 megaspores. The female gametophyte. In a majority of flowering plants, one of the megaspores is functional while the other 3 degenerate. Only the functional megaspores develop into the female gametophyte, which actually forms the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. Formation of embryo sac. The nucleus of the functional megaspore divides mitotically to form 2 nuclei, which moves to the opposite poles, forming the 2 nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the formation of the 4 nucleate and later the 8 nucleate stages of the embryo sac. After the 8th nucleate stage of the cell walls are laid down, leading to the organization of a typical female gametophyte or the embryo sac. 6 or 8 nuclei are surrounded by cell walls and are organized into cells. The remaining 2 nuclei, called the polar nuclei, are situated below the egg apparatus in the large central cell. Development of the embryo sac. As we have said already, now we see diagrammatically how the embryo sac is developed. This is the egg cell and this is the polar nucleus. Now, embryo sac with the haploid nuclei is formed, undergoing mitosis, and the embryo sac with the n number of chromosomes. Finally, this is the embryo sac or the MMC or the megaspore mother cell, which has 2n number of chromosomes because it had already undergone the meiotic division. It has a well-defined nucleus and integument, and this is the wall of the ovary. This is the ovary and the ovule. This diploid cell divides by meiosis once and mitosis thrice to give 2 polar nuclei, and the embryo sac is formed containing the egg cell. The carpel with mature ovary then develops. This is the stigma, style, ovary and the ovule. The ovule has the nucellus, polar nuclei, the egg cells, the integument, and together they comprise of the female gametes. Types of ovules: orthotropous, the micropyle, the chalaza and the funicle are in a straight line. This is the most primitive type of ovule. Piper, polygonum and cycas are the examples. Anatropous, in which the ovule turns at 180-degree angle. It is an inverted ovule. The micropyle lies close to the hilum or at side of the hilum, found in 80% of the angiosperm families. The campylotropous ovule is the one which is curved more or less at right angles to the funicle. The micropylar end is bent down slightly, example in the members of the Leguminosae and the Cruciferae. Hemianthotropous is wherein the ovule turns at a 90-degree angle upon the funicle or body of ovule and is at right angle to the funicle. Example is the ranunculus. Amphitropous ovule as well as the embryo sac is curved like a horseshoe. Lemna, poppy and alisma are the examples. The circinotropous ovule is the one in which the ovule turns at more than 360 degrees of an angle, so the funicle becomes coiled around the ovule. Examples are Opuntia and Plumbageniaceae. Pollination. Transfer of pollen grains, which are shed from the anther to the stigma of a pistil, is termed as pollination. It is of 2 types, self-pollination or autogamy. If the pollen grains are transferred from anther to the stigma of the same flower, the process is self-pollination or autogamy. Contrivances for self-pollination. Bisexuality. Flowers should be bisexual or hermaphrodite. Example is Cartharanthus. Homogamy. The male and the female reproductive parts in a bisexual flower which may mature at the same time. Example is the Mirabilis jalapa. Cleistogamy. Sometimes the bisexual flowers remain closed and never open. Such flowers are known as cleistogamous. Example is Camelina bengalensis, Viola, Oxalis and Arachis. Bud pollination. When the self-pollination occurs in the bud stage before the opening of flowers, example is pisum, wheat or rice. Cross-pollination. When the pollen grains are transferred from the anther to the stigma of the flower of another plant of the same or different species, it is known as cross-pollination or allogamy. Types of cross-pollination. Geitonogamy. Pollination takes place between the two flowers of the same plant. Genetically self-pollination, but ecologically it is cross-pollination. Dichogamy. In bisexual flowers, the two sexes mature at different times. When the anther matures first, it is known as protandry. Examples are the sunflower and cotton. When gynoecium matures first, it is called as protogyny. Example is ficus, aristolochia and magnolia. Heterostyly. Flowers are dimorphic with regard to the length of the style, thus facilitate cross-pollination. Example is primula or jasmine. Hercogamy is the presence of natural and physical barrier between the androecium and gynoecium. Example is gloriosa salvia. In Calotropis, gynoecium is fused with pollinium and forms the gynoecium. Self-sterility or incompatibility. Due to physiological or genetic reasons, the pollens fail to germinate on its own stigma. Example is tobacco. Entomophily, that is pollination by insects.